in listen-only mode.
Good morning, and welcome to the second webinar in our RA Community Phase series. We're pleased to have all of you here today. So we have a couple of things that we'll be doing today. The first is going to be a presentation and discussion about feature selection. So as promised, we have Robert Plenge and Eli Stahl here. They are responsible for the collection of the discovery data set and the genotyping that happened in the test data set as well, and have spent more time than anyone else in the world analyzing these data and thinking about this problem. And so they're the perfect people to be talking to you about their perspectives on feature selection. So what we're going to do is we're going to have them present, and then we're going to open it up for questions. And so any questions you may have about feature selection for them or comments in general, we'll take at that time. And then after that, we're going to introduce the groups as they stand. So everybody's formed into a group, and we'll just take a look at that, get a little introduction to each of the groups. Okay, so without further ado, I'm going to turn it over to Robert, who's going to get us started.
Okay. Can I assume everyone can hear me okay? Maybe I'll just give you just very quickly just a background on me and how I got involved in the RA Responder Dream Challenge. So working with Eli and a number of other people, we had done a lot of work on genetics, not only of risk of rheumatoid arthritis, but response to therapy. And we had done a lot of modeling of the data, but realized that there's probably much more out there that could be done. And that's how this then came to be. So I'm really pleased that everyone has gotten so involved in the analysis of the data set and how we then have been able to make additional data available to hopefully better interpret the complex polygenic architecture of this particular phenotype. As one disclaimer, I actually started this project when I was in academics, and I actually now work at Merck doing something similar, although not specifically on rheumatoid arthritis and in response to anti-TNF therapy. So maybe if we go to the next slide. So just I want to provide a bit of an overview of the clinical indication, and I think everyone probably realizes this by now, but just to make sure that everyone is clear. So once a patient is diagnosed with rheumatoid arthritis, the general progression of treatment includes starting on different types of anti-inflammatories, and including methotrexate, often steroids and non-steroidal anti-inflammatory drugs. But many people, probably 2/3 or more, will fail to respond to that simple combination of drugs. And then the first line biologic therapy is anti-TNF therapy. And this drug is, and this class of drugs are remarkably effective, but as is often the case with these complex diseases and medications, response is incomplete. And so, the numbers vary from study to study, but in general, about 30% of patients will fail to respond adequately to anti-TNF therapy. And by adequately, I mean enter into extremely low disease activity or even remission. And if this were the only medication on the market, maybe there wouldn't be a need for predictors, but there are other options that are out there, and some of them are shown, at least biologically, on the slide on the right. And so having a genetic predictor to guide treatment would be very useful. But I think probably equally important, and this will come up in the discussion today, I believe, is that new therapies are actually needed. And if you could understand a biologic basis as to why people fail to respond, or I guess the flip side of that is why they do respond, then you could actually develop therapies that are targeted against those refractory pathways. So not only is there value in terms of predicting who will respond, but there's probably as much, if not more, value in understanding the biologic pathways to guide new therapies and the development of new drugs to treat refractory RA. Next slide. Let's see, was there one before that, or is this, I saw it skipped quickly? Okay, good. Okay, so Eli, I was going to speak just a few minutes on this, but let me know when you want to jump in terms of talking about some of the results. I know that you did a lot of the analysis here. And so when we initially started the project now a few years ago, and you can see the publication shown here from PLOS Genetics, is effectively what we're calling the discovery data set for the DREAM Challenge. Was published, but all we did was single SNP analysis. And so we got some hints of some single variants that look like they were predicting response to anti-TNF therapy. And there's the top SNP is shown here for a response to one of the 3 drugs that were included in this study, etanercept, which is a soluble receptor and a slightly different mechanism than the monoclonal antibodies. And I think what was interesting about this was not only was it a result that approached genome-wide significance, but the gene itself was a good biological candidate gene, CD84, and there was a correlation between expression of this gene and disease activity, and this was independent of genotype. So there was some correlation with the gene itself and disease activity, and that's shown in the middle. And then on the right, if you standardize CD84 expression, you can see a bit more detail around the correlation with disease activity score. So it suggested that maybe there is something when you look at this at the level of single SNPs, but I think the real challenge was there was no obvious single variant that was predicting response. Is there something more sophisticated that we could actually do with the data set, thinking about polygenic response? Maybe if you go to the next slide. And so that then led to this particular challenge. And if you think about this in the context of everything that's going on in complex traits, and I think this is pretty safe to say that this particular phenotype of response to anti-TNF therapy is a complex trait, that most of these phenotypes are highly polygenic and highly complex, which would suggest that there is an underlying polygenic architecture to this phenotype as well. So as you can see on this slide, then the challenge is there anything else? Is there anything interesting about predictive features, and how could we build better genetic models incorporating clinical data into those genetic models, and could that help us with predicting response to therapy, but then also providing insight into the underlying biological processes. So if you look at B and C, the next challenge here is, do predictive features suggest biomarkers that could be more predictive? What types of biomarkers may enhance prediction? And then the future ultimately would be, can biologists and prediction modelers somehow work together to make better progress in terms of tackling this complex and important clinical phenotype? So next slide. Let's see. So Laura, did you want to talk about this, or Eli?
Yeah, sorry, I had to go off mute. Yeah, so
maybe I can stop there. Yeah.
Sure. Should we pass it over to Eli?
Or I can stop and see if there are any questions first. We could take a few quick questions on the background, or we could just go directly to the next part of the presentation.
Absolutely, we can take some questions at this point. So if you have a question, you can raise your hand, or you can type it into the question box. Are there any questions at this point?
Okay.
Yeah. Okay, so let's move on to Eli. Do you want to take it from here, Eli?
Sure.
Great.
This is Eli Stahl, and I've been involved. I was involved in the first study, publishing the discovery data and conducting a genome-wide association study of TNF treatment response. So now we've been through the team phase of this challenge, and we can see these results for sub-challenge 1, the correlation between predictors and true delta DAS, the change in disease activity score after treatment on the left, and then the area under the curve for predicting non-responders on the right. And on the right, the vertical red line shows random. On the left, the random correlation would be 0. And so the predictors have done far better than random, of course. But this red diamond on the left shows a clinical data only prediction. In fact, this is based only on the single clinical variable that's most highly correlated with treatment response, which is the baseline disease activity score. So the result here is that while we are doing better, adding the genetics to the clinical data is only a marginal improvement. So moving forward, next slide. So what now? We have this community phase of the challenge. We really need to address the question, is there a meaningful contribution of genetic signal to the prediction? And are the community groups and we as a community able to improve on these predictions by combining model strategies? And can we improve on response by incorporating prior knowledge into feature selection? So that's what this webinar is about. And next slide, please. So I think Robert's introduction really got across that, you know, as rheumatoid arthritis geneticists, we're looking at this challenge really as a two-way street. We'd like to learn about the biology, and I think to make progress clinically, there will be some learning about biology that needs to come out of this. But also, we believe that the biology can inform the predictive modeling, and I think we all share this belief. In the challenge, and so this is the star. I'm going to be talking about the arrow in this direction. This is the challenge, though. How do we best utilize biology and external data as part of this community phase of developing a better clinical plus genetic predictor? Next slide, please. So for biology into predictive modeling, I'm going to go through this outline. Point A here, I'm going to present some of the analyses, the polygenic analyses, where we did a sort of within sample proportion of phenotypic variance explained type of analyses, which really suggests that there is genetic signal in the data, but that it comes from the combined effect of many of the SNPs, and so the implication being that more features is going to be needed to derive a better predictor. Of course, as many of the team challenge summary statements and presentations in the last webinar pointed out, that's very challenging when you add features dealing with overfitting, and then when you go to out-of-sample prediction, it doesn't improve things. But moving on to point B and C, I'm going to talk about some of the external biological data and genomic data that are available that we could potentially use to select features or possibly upweight features that have some external evidence for either functionality or specific function that is relevant to the phenotype that we're interested in. So I sort of dichotomized this into point B, SNP level external information, which includes annotation as well as genetic evidence. And then I'm going to make an aside here, these points with asterisks at the end, just to review the ancillary data that are available as part of the challenge, which I think are underutilized so far, and then jump back up to point C, thinking again more about publicly available data, but potentially a strategy that could be of interest to use pathways or networks to inform feature selection. Next slide, please. Really the analyses that gave us a hint that there was genetic signal in the data, and that really sort of inspired the challenge, were these linear mixed model SNP heritability estimation analyses. And we used this software GCTA that was developed by Michael Goddard and Peter Vischer and colleagues, essentially asking if there's a correlation between relatedness between the samples and phenotypic similarity, really where all the samples are unrelated in this kind of GWAS data. So the correlation would be weak, but significant, and this has been shown for a lot of complex traits with at least some degree of heritability, that the common variants in aggregate explain a lot of the phenotypic variance. Next slide, please. So applied to the discovery data, we found some significant SNP heritability results. And in particular, in the middle column of results, SNP heritability, and then the right-hand column, the p-value for these estimates being significantly different than 0. The top row of results was the results for all patients, the discovery data as a whole, finding about 20% of phenotypic variance explained by all the common SNPs together. And then on the bottom row, combining patients treated by the monoclonal antibodies, infliximab or adalimumab, we found a higher level of SNP heritability, 36% phenotypic variance explained by all the SNPs and a greater significance. So these results really suggested to us that that there is genetic signal in the data. But it's worth pointing out that these results are derived under a model in which literally every SNP in the data has an effect. And there's a normal distribution of effect sizes, and these SNP heritabilities are the variants of those effect sizes across all SNPs. So certainly, if this is the kind of signal that's in the data, it requires many features to sort of pull that out into potentially a predictive model. And I don't mean to suggest that we should be trying to attain these kinds of phenotypic variance explained in an out-of-sample prediction context. And really, it's because of the finite nature of the discovery data, we don't expect to be able to differentiate between larger effect and smaller effect SNPs that well. But certainly the implication, to me at least, is that a larger number of features, if we can extract them, could boost the genetic signal in prediction. Next slide, please. So just to point out that this is a very active area of research, and besides the Goddard and Vischer group developing these kinds of models, Matthew Stevens' group at the University of Chicago, and David Balding's group, Doug Speed and David Balding at University College London, are developing these kind of linear mixed model estimators of phenotypic variance explained and predictors, which are in this family of BLUP predictors from the linear mixed models. And in particular, on the bottom right here, this paper I just found in Genome Research. It advanced online, so it's in press still. This multi-BLUP prediction software could be something that one of the groups could experiment with. Next slide, please. So now back to the outline. We have this polygenic evidence for genetic effects in the data. And what are some of the information that are either publicly available or available as part of the challenge that we could think about using to sort of, overcome our finite discovery data sample size issue and help to somehow extract more features from the data. And at the SNP level, there's been a lot of studies, not in treatment response and not in these data, but across complex traits, showing that annotations and genetic function of SNPs is important. So next slide, please. Oh, I'm sorry, I forgot about this. Just to mention that, you know, we know and we're happy that that the participants in the challenge, of course, are aware of the importance of biological knowledge in feature selection, and basically all of the team challenge predictors involved some external data in feature selection, usually up front, and so again, and typically leading to a relatively modest number of features that were used to develop predictive models. So again, this is part of the challenge. Can we bring more features in, even in an experimental sense, to see if they could potentially help prediction. So next slide, please. So again, now the SNP level external information having relevance across complex traits. So on the top left here, this paper in PLOS Genetics last year, showing that if you just take GWAS results from any number of traits, and I think the results here shown for schizophrenia, if you separate SNPs according to their genic annotation, just intergenic versus genic, exonic, intronic, or UTR, the result here actually, and this is across several different complex traits, 5' UTR was the top signal, implying that, you know, there are regulatory elements typically in 5' UTRs in genes, and these are enriched for significant, meaning low p-value signals of association in GWAS. And so the implication here again would be that this kind of annotation, you know, brings with it sort of a prior for SNPs being functional. So given the same p-value, perhaps, from the discovery data GWAS that we would all do as the first step in feature selection, if we focus on, say, SNPs with a functional annotation like 5' UTR, given the same p-value, something with that annotation would have a higher probability of being truly associated and therefore having some predictive value compared with, certainly here, intergenic annotation. And then turning to a different kind of functional evidence in other data sets where we have genotype and gene expression data, EQTL analysis. So on the top right, now there's been many papers from this group led by Nancy Cox at University of Chicago showing that SNPs that are EQTLs, so where the genotype of the SNP has an effect on gene expression level, these are more likely to be associated with phenotypes. And here the analysis is of all of the validated associations in the GWAS catalog, showing that they're enriched for being EQTLs. And then finally, one other form of sort of annotation that's derived from functional genomic data. Here the histone marks, which typically are falling in promoters or enhancers, regulatory regions of genes, in the same kind of analysis as the short paper on the top left. This is also for schizophrenia, a paper that I'm involved with by Roussos, submitted, showing that EQTLs or eSNPs here, but also SNPs that lie in these histone marks are enriched for statistically significant signals of association. Okay, next slide, please. So, you know, there's lots of EQTL data that are publicly available. We have these browsers. I've used the University of Chicago browser a lot. This is an interesting browser, genenetwork.nl, that was put together by Luda Frank, and it's based on a meta-analysis of many studies involving a total number of samples of like 8,000 samples. So a lot of power potentially to identify EQTLs and either upweight features or focus on putatively functional SNPs as part of feature selection. Other resources as well, and then the ENCODE and epigenomics roadmap projects have those other annotations, DNA hypersensitivity and histone marks that could be useful as well. Next slide, please. Okay, so returning to the outline, and now in this kind of realm of genetic function, we do have an EQTL data set that's available as part of this challenge, but we have a couple of other data sets that are available as part of the challenge as well. Next slide, please. So one of them is an experiment that was done actually by Robert some years ago, measuring secreted TNF in the HapMap cell lines. So the HapMap cell lines, as many of you will know, are publicly available genotype data used very often as a reference panel and as for population genetic analyses. But we have TNF alpha secretion measurements for these 270 HapMap cell lines. And so this is a quantitative phenotype. And again, the genotypes are publicly available from HapMap. And this could be used for an essentially EQTL analysis, but looking at SNPs that are involved in the level of TNF alpha, which is the target of the drugs here. So very relevant phenotype. So if one of the groups, could invest the resources to do this genetic analysis, could be helpful in feature selection. Next slide, please.
And maybe Eli could just jump in real quickly.
Yeah, please.
One of the things about that particular data set, it was
previous slide.
Yeah, go back just to slide 19. So I mean, this is something that we generated a number of years ago as part of a much larger HapMap cell line project. And we measured a number of different secreted cytokines at a resting state. And this was one that was probably one of the more robust measurements in terms of reproducibility across, not just duplicate samples on the same day, but samples over and cell lines that were measured and across many months apart. And we actually have never really, other than doing just a basic GWAS on TNF levels, we haven't really dived more deeply into some of the polygenic methods that Eli described earlier. So I think one very simple thing that could potentially be done here, if there's interest, is to say, what about building a polygenic predictor of TNF levels, and then asking how well does that actually predict response to anti-TNF therapy, or even some of the sub-features like disease activity at baseline, et cetera. So I think this is a data set that really has not, even though it's been out there for a while, has not been used at all, and potentially could be quite valuable.
Yeah. Next slide, please. Okay, I think that the ancillary data set that's been actually the biggest investment of resources on the part of the challenge organizers and gotten the most publicity in terms of being available as ancillary data is this arthritis internet registry data, which is RNA-seq data, and the genotype data for these 60 patients available on Synapse. And so two potential analyses that could be done that would be relevant to feature selection here. One is the EQTL analysis, so looking for SNPs that are associated with gene expression and potentially also associated with disease activity, where we have 30 patients with high disease activity and 30 with low. And then also there is just the idea of doing a differential expression, asking what genes are differentially expressed between high and low disease activity patients. And there is actually some results from an analysis of that that are available on Synapse. Next slide, please. And so now straying a little bit from the idea of genetic information and EQTLs, more in this realm of differential expression. We have curated a list of the publicly available gene expression profiling data sets from GEO and listed them here, again, on the Synapse webpage for the challenge. And these are experiments that were specifically geared towards treatment response, anti-TNF treatment response in rheumatoid arthritis. So these, again, differential expression and pointing to genes that where the SNPs that are in those genes, again, for a given level of association with treatment response in the discovery data, one might think they should be upweighted in terms of feature selection for predictive modeling. Next slide, please. Okay, so now finally to point C here, again, following up on this idea of gene-level external information and thinking about pathways and networks a little bit. And one interesting idea that we've started to play around with a little bit in the kind of linear mixed model SNP heritability analyses is the idea of using networks or gene sets to expand a limited set of features to a larger set of features and asking if that seems to be pointing to some of the signal in the data. So next slide, please. Typically, what we've used for these kinds of analyses are there is this import reference data from NIH that has pathways, including TNF pathway, but also just a list of all immune-related genes, a list of all cytokine signaling genes, and so on. And then protein-protein interaction data. I'm used most this InWeb database that was developed by Casper Lodge, but there's a lot of other databases that are out there. And then there's been many analyses focusing on co-expression data to build networks. And again, I'm pointing out this webpage genenetwork.nl. That includes some tools for seeding a network with some genes of interest, like you might do for if you had it starting out with a modest set of features that you selected, and then expanding that out based on co-expression and getting a larger set of genes to that you could look at SNPs that map to those genes as a larger feature set. And the ImGen and Novartis Gene Expression Atlas, maybe some of you are familiar with, are also larger scale multi-tissue expression profile data sets where you could look at co-expression, at correlations to build networks. Next slide. So we've done a little bit of this using the SNP heritability type analysis in the discovery data and not finding any home runs here to point out, but if you look at the first row of results, these are the whole genome results that I showed in a previous slide. About 20% variance explained, 18% variance explained for all samples, 36% variance explained for the patients treated with the monoclonal antibodies, infliximab plus adalimumab. And if we look at the rows further down then, here we are focusing on SNPs that map to genes that are in these particular gene sets where we've used those external data sets to do SNP selection to build these variance component models. And so, the first bold result set is in the far right column, and the third row of results down, 0.21 variance explained. So here it's about 2/3 of the whole genome variance explained, 0.21 versus 0.36 at the top. And then if you look over on the left at either the percent of SNPs or the percent of megabases of the genome, it is also about 2/3 of the genome. So here 2/3 of the genome explains 2/3 of the SNP heritability. So not showing necessarily any concentration of the genetic effect that we think could be important for feature selection. But we do see some intriguing results if we look at the results that I've made bold and italics. So the next row down, but for all samples here, 0.05 heritability is about a quarter or more than a quarter of the total SNP heritability, but coming from 0.11% of SNPs, so 10% of the genome giving 25% of the heritability, of the SNP heritability. And this is the TNF, the target of these drugs, expanded by direct connections in the InWeb protein-protein interaction database, and then further expanded by selecting genes that have a high correlation in the co-expressed networks from that Netherlands website, so leading to 333 genes in that gene set. And then in the last row, the result that's bold and italics on the far right, here's an approach that could be interesting as part of feature selection. So in the discovery data GWAS that we performed for infliximab plus adalimumab together, we found a suggestive association on chromosome 7. And then if we expand that out by protein-protein interaction and look at all the SNPs mapping to 39 genes that interact directly with the gene that's closest to that chromosome 7 top hit, we can see the result is modest, 3% of heritability. It's about 10% perhaps of the total SNP heritability of the genome, but it's coming from only 2% of the genome. So that's kind of the idea behind sort of the concentration of the genetic effect. Can we come up with a set of features that shows a more robust genetic signal? Next slide, please.
Actually, Eli, there's a couple of questions that I think might be appropriate right now. So
sure, yeah.
Read those out to you. The first is, can you just give a brief explanation of what heritability is?
So literally, heritability is the phenotypic variance explained by all of the SNPs in aggregate under this model, this linear mixed model, where we have a random effect for SNPs in the data where they are assumed to follow a normal distribution, and then the parameter is the variance of the effect size, the variance of this normal distribution. So the way, so we think in quantitative genetics, we think of this as the heritability, which is the genetic variance, the additive genetic variance under a model where all the SNPs have an effect. Many or most are very close to zero, but all the SNPs have an effect, and their effects are additive in terms of the phenotypic variance explained. And then the way operationally that this is carried out is by creating a kinship matrix based on all the SNPs, or in these pathway results, where the slide is showing now, based on a set of SNPs that represents some sort of partition of the genome. So this covariance matrix, which is based on the genotypes, kinship matrix, we ask whether that covariance, the genotypic covariance, is correlated with the phenotypic covariance.
Thanks.
And those papers that were in that previous slide where I was showing a couple of the figures from those methodology papers, so those papers are there.
Great. The next question is, when you do these heritability estimates, do you control for baseline DAS as a covariate?
Yes. Yeah. We always have baseline DAS as a covariate, and that's why it's referred to as a mixed model, where you have a fixed effect for any number of covariates, and we typically have included baseline DAS and some of the principal components that we think represent ancestry. And also, these are typically done combining the different studies that make up the discovery data with the study covariates as well.
Okay, then the next question is, have you designed any simulation studies that look at the underlying biological model is predetermined, and then use that to test your polygenic models?
That's a good question. I am involved in some of those studies that are ongoing right now. We haven't published on that yet, but the idea of partitioning the genome into different segments it has been forwarded and being acted on by several different groups, and some of those typically involve simulations. I'm pretty sure that particularly those two papers that I showed the figures from Matthew Stevens and Speed and Balding, those have those kinds of simulations.
Great, thanks. All right, let's move on. Are you ready for your next slide?
Yeah, we're pretty much done here, I think. So, here's a picture of a pathway, moving into the light, and just making use of this sort of pathway reference where we've been talking about gene sets or pathways, but really what this represents is, in the community phase, if this project has a path forward that's going to lead to significant results and significant insights. Next slide, please.
Is that, hey Eli, just a
yes.
quick, is that a photo from your backyard in Brooklyn?
No, it could be Central Park maybe though, but this happens not to be.
Yeah. Okay.
It's debatable whether that kind of pathway into light is truth here that we're looking at in this challenge, but I think we are all in this community phase to work together to make some representation of the reality, and if we can come up with something like depicted here, I think we'll be doing really well.
Great, thanks. All right, so at this time, I'd like to open the floor for questions. You can, as I said, either raise your hand or just type your question directly in. Okay, so the next question typed in is, do you have any biological reasoning as to why delta DAS is correlated with baseline DAS? Robert, will you take that one?
Yeah. So I think that when patients come into the clinic, maybe I can just say just a few kind of general comments. So when a patient comes into the clinic and they're seen as having very high disease activity, what goes into that score? And I don't know if this has actually been discussed before, but it's a composite index which includes a physician exam, which includes the number of tender and swollen joints. It includes a kind of an subjective feeling of how the patient is doing on a visual analog scale, and it also includes a marker of inflammation like ESR or CRP. So you can put those into a composite score and get this disease activity score. So if a patient comes in with very high disease activity, there is more of a chance that disease activity will will change just because there's a greater range when they are started on different medications. I think one reason is probably just a very high disease activity score can correlate with just a greater amount of potential change following any particular therapy. So that's one reason. I think that there are lots of other potential clinical or biological explanations, which could include maybe a patient with a particular type of rheumatoid arthritis will have more of systemic inflammation as measured by the blood biomarkers versus clinical inflammation as measured by the clinical exam, and that might actually feed into how and why there's a correlation with change in disease activity over time. So those are, I think, some potential explanations as to why there might be that correlation.
Okay, so then the next question is about SNPs related to RA. Why is it that they can be predictive, but we don't get signal in the GWAS study?
So I think is this correlation of so if you take the risk alleles, the alleles that are associated with the risk of RA, do those same alleles correlate with the response to therapy? Is that the question?
I think what might be helpful for this question is actually to get the person who was asking it on the line. So, Lou, yep, you raised your hand. I'm gonna unmute you so you can ask your question verbally. All right, you're unmuted.
Okay, so one phenomenon that we discovered is that we get lots of SNPs from the literature, and then they are really predictive. But then when we are doing GWAS using the delta DS as a phenotype, then for those SNPs from the literature, they don't have a high p-value. That means that they are not predictive. They are not associated with a response. So we don't. Why?
So you say SNPs from the literature, you're talking about SNPs that are associated with the risk of RA, or SNPs being anything associated with autoimmunity or CRP or I don't know, complex traits?
I think it is with RA risk SNPs.
And so those RA risk SNPs are correlated with baseline disease activity, but not correlated with change in disease activity?
Yeah.
Okay.
No. Sorry, they are from okay. So they are from previous some from the literature. There are some SNPs that predict that is found out by using GWAS, using a different phenotype, like whether the patient is whether the patient whether this is an RA patient or not a RA patient, and then using that kind of phenotype. But then in this studies, in this GWAS studies, like whether the patient is responding to the drug and or not responding to the drug. I mean, the phenotypes are different, but those SNPs that have low p-values in the literature, but then they have low p-values in this study. Why in the prediction in the team phase we found these SNPs are really useful, but just do not provide enough information. So we are a bit confused about this.
So, but just to make sure I'm clear, so that if you take RA risk variants, that those having non-random distribution in terms of p-values with regards to association of response to anti-TNF therapy, so that you're saying the word predictive, but they have some, either predictive value or non-random distribution in the response to anti-TNF therapy results. Is that right? That's actually, if that's the case, that's actually pretty interesting. We looked at with a smaller set of SNPs, it might have been 20 or 30, kind of at a stage before we had published our most recent study, which had over 100 variants that were associated with risk of RA, and we didn't see much in the way of a non-random distribution. But it might be that if you go to more SNPs in the model and even go below the sub-genome-wide level of significance, that there is a correlation there. And I guess it could make sense because, variants that are associated with the risk of RA are increased in the likelihood of being functional with regards to the immune system. So I don't know, maybe there's something there. Eli, I don't know if you have any thoughts. And then I'm going to apologize. I actually have to go in just about 2 minutes, but I figured I'd at least take a stab at that question before turning it over to Eli.
Yeah, no, Robert's right. I mean, I think that we can say that, the SNPs so these are SNPs associated with risk of RA. We think biologically that they should be related to the treatment response phenotype. I would definitely be interested in that kind of information in a sort of feature selection, as you did. But either their effects on treatment response are not great enough to where they show up in every study, so we don't really see them strongly in the discovery data, but they do have some value in the validation data. Or it's always possible as well that there's some clinical heterogeneity across data sets, across samples, that contributes to the sort of heterogeneity of the genetic result and the predictive value. It seems like sort of par for course for me, but good results, I mean, to have those in.
Okay, thank you very much.
Yeah. Well, Robert, I know you have to go, but we really appreciated having you here to talk with the participants. Thank you.
Well, thank you, and I think that, I guess one just parting comment is I think Eli and I have spent a lot of time and have a very fondness for this particular phenotype, not just scientifically, but I think clinically as well. So I know that a lot of people have spent a lot of time on these data sets, and thank you very, very much, because I think if we can do something with this complex phenotype to get at a better understanding of the genetics and the biology, et cetera, I think it would be really amazing for the scientific community and ultimately, hopefully, for patients as well. So I want to actually thank everyone else. So thank you.
Great, thanks. Okay, so the next question is there any possibility of going back to the training or test cohorts to gain more information about clinical measures such as anti-drug antibodies or autoantibodies? I believe the answer to that is no, at least not for the training data. It might be possible for the test data. I think there might be blood samples collected from the test data. But the training data come from many different sources, and to systematically go back would be untenable. And then has there been any check on correlation between scoring across clinical sites? Okay, so I don't know if Eli, you want to take that question, or maybe that's a question for Sally. What do you think, Eli?
Yeah, I mean, across sites, like things like the correlation between some of the clinical covariates and the primary response phenotype. We did those kind of checks when we were, writing up the GWAS on the data, and we do see some differences. There are also but nothing that's like strongly significantly different between data sets, right? Mm-hmm. When we did the GWAS that's published, we essentially filtered for SNPs that showed like a strong heterogeneity between data sets in terms of the genetic association with treatment response, and thinking that would primarily reflect issues with the data across sites. And maybe having looked at the data for the challenge, Sally, you have more to say?
Yeah, I think the question is more specifically, when scoring these models, are there trends across the data came from
I see, yeah.
different clinical sites in terms of ability to predict? Did you happen to look at that, Sally?
Well, that only pertains to the leaderboard data, and so we haven't really done any in-depth analysis on on the leaderboard submissions simply because there were so many of them. So
yeah, and I mentioned this in the email last week, but I'll mention it verbally as well, that we do intend to release the leaderboard data at the end of this session, or at the end of this phase, to give all of you a couple of weeks with a slightly extended training set, so you can incorporate that into your models. All right, so that's all the questions that have been written in. If you have additional questions while we proceed, please continue to write them in or raise your hand. Let's move on to the introduction of the groups for the community phase. So, oops. Let me, I'm going to start with, let's see. I'm going to start with Team GSTM. Who is presenting on behalf of GSTN? Please raise your hand. Ah, Lou, excellent. All right, I'm going to unmute you so you can introduce your team. There you go.
Hello everyone. Good evening. This is Juni from Finland. Okay, so the group contains 4 teams. That is Guan Lab, SBI Lab, and Tracy, and MI. So we take the capital letters from each team, and then that's the team name, the group name. Okay. So next slide, please. Since we think that the development of the predictive model is can be majorly divided into 2 parts. So it's first the SNP selection, and then the prediction model. So these 2 parts are relatively independent. So what we think is that the 4 teams still working on their own models and SNP sets, and try to improve based on the previous method and the SNP sets. But every week we will share the SNP sets, and also new improvement about the method, and then we'll report the progress and new results among all the teams. Also, if one team is interested in SNP sets from another team, or they are interested in modifying the method or understanding the method of another team, then they can contact individually. So there will be lots of underlying communication here. So these 4 bars just means that every horizontal bars means every team develops their own method, and then each week there will be exchange of the information, and then this will be group among all the groups. And then in the final phase, we will compare and combine the different predictions, and then decide which final submission we are going to we are going to provide. Next slide, please. And yeah, so these are more details because the previous is a graph. So now, so basically we share the SNPs, and we share new ideas and report progresses and the improvements. So we have a Dropbox folder that every team put their data, code, and report, and progress, something like that, or in a Dropbox folder. And we will try to help other teams to run one team's code. So a team, if there is a request, it will always be solved by the corresponding team. And then also we agree that if other team if it's difficult to run other teams' code, then we can request the team to provide help for running the method, or of course modifying the simple modification of the method, or just generate some kind of SNP set if that team has such an expertise. And we are trying to use a synergy to develop an ensemble method that can combine all different methods we hope that this method will predict better than any individual method. Yeah, so probably that's it. Thank you.
Excellent. Thanks very much. Okay, so the next group is a combination of 3 of the other teams, and it looks like Matthias has his hand raised, so I am going to assume that he's going to do the introduction. Matthias, I am unmuting you. Hello.
Yeah, thank you. Last week, Team Linked Open Data and Team Outliers had a teleconference, and we decided to form a supergroup. And quite recently, we were also joined by Team STSI. And since this is a quite recent development, we could not really yet have a joint teleconference because we are also spread over so many time zones. We will expect to do this tomorrow or the day after, which means that today we can't really present a common strategy shared by our newly founded group. But I think we will be able to present something very soon. So basically, I think the work will be shared so that Team Linked Open Data will specialize on feature selection based on available biomedical data sets, while the other 2 teams, Outliers and STSI, will focus more on the machine learning part based on those features selected.
Well, great. We look forward to seeing what you guys come up with and looking at how you go about this. So thank you. Okay, and so then the last group, as you all know, is Lucia, who is working on a clinical model. So let me find Victor. Okay, I'm going to unmute Victor so he can. All right, you're unmuted.
Hello.
Hello.
So, can you put the slides? Yes. Just go to the next slide, please. Okay, thanks. So, as you know, we have been sending emails to everybody that we actually we are going to focus is in validate that actually we found information in SNPs. For that, I mean, we're going to work in developing models only with clinical, but we would like also that all of you will test your models also using only the clinical features, so using exactly the same model. If you use, linear regression, then a linear regression only with the clinical features. But also using random selected features. I think that these 2 things are not much work and are going to provide us with confidence if we find a method that works and some SNPs that actually works. So if you go to the next slide, please. So yes, besides that, we're going to also be going testing models, and we want to share all the results that we're getting with all of you. So we're going to still sharing all the information with everybody, so everybody can use it. And we still like it that you also share information with us. And our objective actually is not to win everybody. I mean, we hope that we don't, because I mean that will be nice because we'll find some information SNP, so all we do actually matters. So for that, we will actually like a lot if you can share information with us, and if you are, and if all that you test, that if you can share it, so it will be great. And also say that some members from MI team are going to collaborate with us. That's what is confirmed. And if anybody out there wants also to actively collaborate in testing clinical models, just let us know, and we'll be really glad to collaborate with you. So.
Excellent. I think that this is a really important part of this phase, the idea that we need to really take a standardized approach to looking at clinical predictions to be able to properly assess whether the genetics are contributing in a meaningful way or not. And so I encourage all of you to work with Victor and his team because I think it'll help all of us answer this question. So I'm excited to have all of you developing groups and thinking through your strategies. We're going to have these webinars every 2 weeks throughout August. I think this format works pretty well. We don't actually have a presentation set up for the next webinar because we're not sure what would be useful for all of you. If there's something useful for us to be able to provide for you in terms of information. So if you have any ideas or requests, please send them along to me, and we can set up a presentation if you do find it will be useful. But otherwise, we will have internal presentations from each of our groups and discussions. Victor, I see that your hand's raised. Do you have a question?
No, sorry.
Oh, okay.
Sorry.
Fine. Great. So now I'd like to turn it over to Gustavo to say a few words to wrap us up. Oh, wait, before I do that, sorry, there is one more question. And that is, I think, a question for Sally. The question is, should we filter SNPs based on their info score? And if so, what is an appropriate threshold?
Yeah, the info score is telling you how well the SNP is being imputed. So it's probably a good idea to do some filtering based on info scores, just because adding SNPs with lower info scores is likely just going to be adding noise, even if they're predictive from within the training data set. The question of what threshold you use is a little bit arbitrary. I think, for GWAS, people tend to use things around 0.7, 0.8, 0.9 if you really want to be stringent. But it is a good idea to put some limits on info scores.
Great, thank you. Okay, Gustavo, I'd like to turn it over to you now to say a few words and wrap us up. You are unmuted from my end, although it looks like you're still muted from your end.
Now, you can hear me?
Yes.
Okay, great. First of all, I wanted to share, the same feeling that Lara expressed, that it's so great to see that, in a way, for us, it's a dream come true, really, to see that competitors become collaborators, because we think that there is after having developed your own thinking about how to solve these problems, there is great opportunity for us to equalize our understanding, see where we don't understand, and push in that direction. And in my following this challenge from the organizing side, it's clear that there are still the usual questions as to how are we going to figure out what SNPs are most responsible to or predictive of the delta dash, the treatment outcome. This seems to be something that everybody is trying to figure out. It's important what Victor said, that let's not confuse the little signal with the signal that we can get from the same number of SNPs if they are random. I think that's an important lesson that we have learned in the field of genomics in general. And but most importantly, I think that beyond the 2 or 3 groups that have been formed, don't think of you as competitors, even though there is always something there. Think of who can try to answer your question, when you have a bottleneck amongst the people that have different expertise from you, because that's in a way what this phase aims at. And finally, to say that we wish you all the best luck to solve these important problems. We hope that if we know how to learn how to solve it here, it will not only help RA patients, but also probably other patients in other areas. And also, thank you for being the first group that really took on the challenge of collaborating beyond the team phase. For us, this is we are learning how to organize this phase as much as you are learning about the biology and the data analysis for this. And to finalize, thank you, Eli and Robert already left, but we wanted to thank you them as well, because getting the biology and genetics a point of view is essential in order to not just crunch numbers, but really try to understand what's going on at the biological level. So with that, thank you very much. And Lara, back to you.
Great, thanks, Gustavo. And yeah, thanks all of you for joining us and for all the effort you've put into this. And we'll talk again in 2 weeks. And in the meantime, if you have any questions that you'd like to ask of Eli or Robert or Sally or any of us, please send them to the Google group, and we will answer them there so that everybody can see the answers, so everybody gets the same information. All right, thanks very much. Have a good day.
All right, thank you.